का question है. I have a question. Okay, I am recording the session from here on. Let's start with a formal class of molecular immunology. Okay, the question from the video which I have shared with you people was that you people know that red blood cells, the life of the red blood cells is like 3 months or so.
Yes, sir.
Why? आप मुझे बताएंगे why? क्यों ऐसा होता है? Red blood cells
Because they don't have a nucleus.
अगर किसी का न्यूक्लियस ना हो तो वो एक खास टाइम के बाद फोर्म
They cannot divide and they cannot.
Okay, it's a good question. I am leaving it for discussion, and I will talk on about this thing later on. I will going to share my screen, and you should be able to see the lectures which we are should be able to see the slides which we are studying. And here we go. This is like we are trying to study the innate immune system. We started with the hematopoiesis. We started with our understanding about neutrophils. We studied about the macrophages and their origin and the reason why they are different in different cells. We have gone through all these things. We studied about the classical and alternative activation of macrophages. We studied about eosinophils. If you can recall, extracellular killers. We studied about the mast cells. You can relate it with histamine, eye redness, basophils, vasodilation, the increased permeability of blood capillaries. I wish you should be able to recall all those things. And today we will going to talk about the concept of antigens and how many different types of antigens are out there. So all set. So let's start with the molecular immunology class of Foreman Christian College University. Anything which can act as a non-self. 100 बातों की एक बात. Anything which can act as a non-self is an antigen. सारी से आप पे खत्म. Whatever our body, whoever, whichever, living or dead, any component, any molecule which can be considered as non-self can exist, might be, can be an antigen. But this is not all for any antigen. There is something more you need to know. If something is non-self, it can act as an antigen. The second level is that it should trigger the immune system. It should trigger and activate the immune system. The immune system should be able to recognize that thing as a non-self and start doing its job, start doing its function. Okay, so antigens should have two characteristics. The first is the non-self, and the second is their ability to trigger an immune response. It is called immunogenicity. They should be immunogenic. They should be able to trigger the immune function. If something is very bad, if something is very non-self, not nonsense, but something is very non-self, that non-self is not able to trigger the immune system. If that non-self is not able to trigger the immune system, remember that thing is not an antigen for that living body, for that living system. And it's maybe a virus is having all sorts of bad things, but that virus is not able to trigger the immune system. That virus is not an antigen anymore. If you ask me that why a virus is more dangerous as compared to the other one, this is the factor which can categorize the badness of a virus. If a virus is very, very bad, but it can be detected very easily by the immune system, it can be dealt. The solution can be created and the virus can be sorted out. If a virus is not very toxic, very dangerous, but it can avoid the immune system, it can hide itself from the immune system. It's not immunogenic. That means that virus will not going to be sensed by the immune system, and there is no way that we can fix or kill that virus. Even it is not very much dangerous. So the concept of antigen is related to the ability of the antigen to trigger the immune system. Now, when I said trigger the immune system, it has two meanings as well. The one of the meaning is that a bad thing enters into the body and it has the immune system or innate immune system, they started considering that thing as a foreign stuff, as a non-self stuff, and try to kill that thing. The second level of immune response is that the body considers that thing as a bad thing and then initiated the cascade of events which leads to the creation of antibodies. If the antibody formation is there, that means it's like a complete action. Like from the very start, by understanding about the bad thing to the very end where the body created the antibodies. If an antigen is able to trigger all that path, that antigen is considered as a complete antigen because it can produce a specific response. It can produce a specific observable reaction. And that antigen is also considered as a proper or a complete antigen. इसको ये एक अच्छी चीज होती है. There are other molecules like haptens. There are certain substances over there which are incapable of inducing antibody formation by themselves. They are not able to induce antibody formation. ये immune system को इतना push नहीं कर सकते that immune system consider those things as that bad, so they will not going to produce any antibodies. Although they are antigens, although they can bind with any antibody, although they can be killed with any XYZ antibody, certain other specific antibody, they can be killed by anybody, but they don't have antibody raised against their names. उनके अपने नाम के ऊपर कोई antibody नहीं बनती है. Such things are called as the haptens. So there are certain molecules which are not acting as a complete antigens. They are just like haptens. तो ये terms जो हैं ये कभी literature में कहीं ना कहीं इस्तेमाल होते हैं. The other things are easier. This will be our first complete online class is quite easy. Antigen, how we can determine the immunogenicity or antigenicity. कोई चीज antigen होएगी कि नहीं होएगी. Something is antigenic or not, or something is more antigenic, or something is less antigenic. Something is highly antigenic, or something is not reasonably high antigenic. There are certain factors which can define those things. It's quite easy, but when you are working as a researcher, or when you are trying to trace or trying to create a project, or trying to hunt down a disease, an infectious disease, or a disease because of antigen, you should know all these things, and you should keep all these things in your head, in your mind, before making your conclusions. A large molecule, naturally, it will going to be highly antigenic. A low molecular size stuff will be less antigenic. It can, it should make sense. अगर एक चीज, if something is a very किसी का size बड़ा है, so it is much easier for the immune cells to detect that stuff. The next is very critical, that is the chemical nature of that thing. If they are made up of proteins and polysaccharide, they will going to be highly antigenic. But remember, those proteins should not be the proteins of the host. Like humans' immune system are very active and can consider anything very easily as an antigen if that thing is made up of proteins unrelated to humans. मसलन एक बैक्टीरिया का प्रोटीन है. A bacterial protein is antigenic because their structure, the protein structure is very different from the proteins we are carrying. So we can consider it very easily as an antigen. Although the proteins are part and parcel of life, but the protein structure of bacteria and viruses and other proteins which are not related to humans, all of them are quite highly antigenic. And especially the polysaccharides. We are not very much good in having lots of polysaccharides which are used by microorganisms. Cell wall plants में होती है, हम उसको जानते हैं, हमारी body भी जानती है. But the cell wall present in the bacteria is different. The cell wall present in the fungus is different. If you remember, chitin, fungus में chitin की होती है. Bacteria में polysaccharide की होती है. So that's why our body is able to distinguish those things very easily. Lipids and nucleic acids are comparatively less antigenic. And this is a very important point. This point is very, very important. That means if a substance is sending a nucleic acid only, you can guess the substance. You can guess the thing, virus. It is difficult for our body to predict from the sequence of a DNA that whether it should be considered as antigen or not. It's not easy. It's less antigenic. So that is why if only nucleic acids are the reason of the bad thing, our body will going to feel more troubled. We're going to work more harder in order to understand the trouble. जी राफिया.
सर मुझे पूछना था कि is there any such disease which is because of the fact that जो invading agent है उसके protein और polysaccharides जो हैं they are similar to human polysaccharides and proteins and उसकी वजह से हमारा immune system is unable to detect them.
There are many. There should be many. I'm not pointing out the polysaccharide thing. There are many diseases which arises because that our immune system failed to understand that thing as a bad thing, as an antigen. So something is very bad, but somehow their structure, their sequence of DNA or their structure of protein is so close to humans that our immune system failed to distinguish those things as a bad thing. Although they are antigens, but if we consider those things as no antigen, our immune system will not respond at all. If we consider them as less antigens, our immune system will respond to these things with a lesser magnitude. So these things matter. Many diseases are because of that thing. And you will see and you will browse in Google. बीमारी के होने के बेशुमार reasons हैं. One of the reason is that our immune system failed to consider something which is bad as bad. Failed to believe on something which is bad as bad. होता है ना आपके भी इर्द गिर्द बहुत सारे antigens होते हैं which you should consider as antigen, but you just consider them as self, very very self, close self. But they are not very close selves. They are supposed to be antigens. They should trigger an immune response in your system. But somehow the proteins they are carrying, they are so close to you. They look very close to you. They look very similar to you. The genes they are carrying, they look very similar to you. So you just started considering them as self. Okay? Sorry about this. Next. So,
sir, I have a question.
जी मरियम शाबाश.
जी सर, मेरा क्वेश्चन है कि जो वैक्सीन्स हैं, what's their chemical nature? Are they less antigenic या highly antigenic होती हैं? Because अगर highly होंगी तो ही they will trigger the response, I guess. The cascade of events that you were talking about.
Yeah, it's a good question. And first thing, let me answer the question. They are made and designed in a way they should be highly antigenic. उनको डिजाइन ऐसे किया जाता है कि उनकी हमारी बॉडी जल्दी उनको पकड़ ले. उनके अंदर ऐसे कंपोनेंट्स जानबूझ के ऐड किए जाते हैं. Certain components are added in a way so that our body can consider it as a bad thing. Now you can ask me whether the vaccine itself is a bad thing. The answer is no. Whether the vaccine will itself is going to kill the body, the answer is no. So that is what we call as a vaccine, something which is not going to kill our cells, but they will trigger the immune response. ये safety है. It is called vaccination. इसको आप
Sir, I have a question.
हाँ राफे, इसको एक मिनट में कंप्लीट कर दूं. इसको आप ऐसे समझ सकते हैं, इसको आप इधर गया मेरा माउस. इसको आप ऐसे समझ सकते हैं. फॉर एग्जाम्पल if you create a virus, you know virus क्या करते हैं जनरली तौर पर. Viruses send their genetic material into a cell which is called as a host cell. उसके सिस्टम को हाईजैक करते हैं, हम उसको पढ़ेंगे, उसको हाईजैक करते हैं. एंड आफ्टर दैट वो क्या करते हैं, the host cell will start producing viral proteins and nucleic acids. And after a certain period of time, host cells की capacity खत्म हो जाती है, resources खत्म हो जाते हैं. And then what will happen? The host cells burst and releases lots of new viruses. Now think about creating a virus. अगर आप एक वायरस बनाएं which is completely a virus, like coronavirus, which is 100% not, which is completely look like a virus, but it simply don't have that special protein which allows the virus to make a hole on the human cell and then pass down the genetic material. सब कुछ है उस वायरस के पास, सिर्फ क्या नहीं है, does not have that enzyme which create that small little hole on the human cell so that the virus can pass down its genetic material. उसके पास सब कुछ है, सिर्फ वो वाला एंजाइम नहीं है जो उसको अलाउ करता है, उसको इस काबिल बनाता है that it can pass down the genetic material or inject its genetic material into the human cell. If I ask you, this virus is an antigen, क्या ये वायरस एक एंटीजन है? You people should answer me yes.
Yes, sir.
Everything. If I ask
Yes.
you, as
Yes,
this
it would be an
virus
antigen.
is going to create a disease,
No, sir.
the answer is
No.
the virus will not going to create a disease because it will not going to transfer its genetic material to the cell. So in that case, such virus will exist in the system, but will not going to create any disease. So our immune system can detect them, and our immune system can start creating the antibody against that virus. Now, when the real virus come after that, जब एक सच्ची मुच्ची का virus आएगा जो completely functional होगा, we already have that antibodies against that similar sort of thing. So this is the concept of vaccination. एक और level भी है vaccination का. In that case हम क्या करते हैं?
Sir,
राफिया मुझे भूल जाएगा, ओके? सवाल भूली ना, संभाल के रखिए, ओके? ये जरूरी बात है जो बता. Another level of vaccination is there, और मैंने ये खुद भी किया हुआ है. I have done it myself, not vaccination but with the virus. If you engineer a virus in such a way that you deleted those proteins which allows that virus to replicate. If you delete those genes in the nucleic acid of the virus, which enables that virus to create proteins for the replication. So in that case, whether that virus can infect the human cells, the answer is yes. Whether the virus will be able to enter into the human cells, the answer is yes. Whether that virus or the viral nucleic acid will be able to capture the human's genome, the answer is yes. The next question, whether that virus will going to produce more viruses out of human cells, the answer is no. It will become impotent, that impotent virus. Now that virus is working like a virus. अगर आप human cells, human body को लगेगा antigen, it will going to enter into the human cells, the answer is yes, in fact. But it will not going to multiply. It will not going to produce more viruses out of the human cell. So that in that case, it is mimicking everything perfectly related to the virus. So our immune system will start fighting against that virus, will start creating the antibodies or start creating the cells against those viruses. Now, since that virus is not replicating, so it is much easier to eradicate those viruses. They will not going to reproduce. They will, if they are 100,000, they will stay as a 100,000 and they will be killed sooner or later. So that is how vaccines are created. These are advanced levels of vaccine creation. जी राफी आपकी बारी है.
सर मुझे पूछना था कि जो कैरेक्टरिस्टिक जो कैटेगराइज करी है कि कौन सी हाईली एंटीजेनिक है या नहीं है, क्या इसका ताल्लुक फॉरेन एजेंट की स्ट्रेटेजी से है?
मुझे आपकी आवाज नहीं आ रही, आपके पास शोर बहुत ज्यादा है. आई एम नॉट गेटिंग योर क्वेश्चन. किसी और ने सुना है? अच्छे से सवाल तो रिपीट कर दे. राफिया ने क्या बोला है? राफिया आप लिख दीजिएगा, आप टाइप कर दीजिएगा. ओके, आप टाइप कर दीजिएगा. लेट मी टेक यू टू द नेक्स्ट thing. Okay, so these are the factors which are defining about the antigenicity of the, and I also want to add one note to you people कि अगर हमारा, वैसे मैंने सुबह सुना है कि जूम वालों ने मीटिंग टाइम बढ़ा दिया है, लेकिन अगर ऐसा नहीं है और हमारी मीटिंग अब्रप्टली खत्म होती है तो you should expect me to send you the link. Okay, the new link. मैं आपको इमीडिएटली आपके व्हाट्सएप ग्रुप पे न्यू लिंक सेंड करूंगा एंड यू कैन ज्वाइन इट. सो आई गॉट द क्वेश्चन, इज एंटीजेनिसिटी रिलेटेड टू द पैथोजेनिसिटी और विरुलेंस ऑफ द इन्वेडिंग एजेंट? द आंसर इज yes, it is related. If you ask me related, the answer is yes. It is not the same thing, but it is related. Okay, the next thing is that we can consider something as an antigen if it is affecting any enzyme which is part of our system, if it is destroying any enzyme, if it is inhibiting any enzyme which is present in our system. If and of course, if it is a non-self, we never found that thing in our human body, that is considered as an antigen as well. Antigen specificity, or how our immune system will see the antigen, it is quite interesting and important, because immune system don't have eyes like us. We can see the person, we can define that person, we can explain based on the height and skin tone and hairs and its structures. We can define a person with our own eyes. The immune system don't have such capacity to see the antigen. We should understand one thing, the first thing, that our immune system consider one antigen and take it as a whole in one picture, or our immune system will recognize the antigen from its different components on the same structure. Let me make it more easier. ये important है. If a protein is an antigen, अगर एक protein antigen है, if there is a protein which is an antigen, name it antigen X. If a protein is an antigen, it's an antigen X. Now that protein is made up of 4 domains. जैसे protein structures different domains से मिलकर बनते हैं, that protein is made up of 4 different domains. Now name all those 4 different domains, like domain A, domain B, domain C, and domain D. There are 4 domains present on that protein. You should ask a very important question, that whether our immune system will consider this protein or this everything as an antigen X, means everything, or considered as antigen A. It should recognize that protein from the one of the domain which is called as A, or from the second domain which is called as B, or the third domain which is called as C, or the fourth domain which is called as D. So our immune system will recognize, will consider, will record that antigen as antigen X, or antigen A, or antigen B, or C, or D. It's a very good question and a very good point. It's an important point. Even if one antigen is carrying 2 different molecular orientations, those 2 can be considered as 2 different antigens. although those antigens are coming from the same protein. Although those antigens are coming from the same protein. यहां से एक बहुत मुश्किल कॉन्सेप्ट है, वो समझ आता है, बट दिस इज नॉट द टाइम टू टीच यू. आपने टर्म सुनी होगी पॉलीक्लोनल एंटीबॉडीज और मोनोक्लोनल एंटीबॉडीज. मैं इसको आपको क्लैरिफाई करना चाहता हूं 100% पत्थर पे लगी की तरह. But let me go through some easy definitions first. Isoantigens, they are the antigens found in some but not all members of the species. They are called as isoantigens. They are present in one member of the species, like blood grouping. आप देखते हैं, some people have the antigen called antigen A. Not every person is carrying the same antigen. Such antigens are called as isoantigens. The other term, autospecificity, autologous or self-antigens. Especially they are not antigenic, they are non-antigenic, but they are very interesting exceptions. They belong to us, but we don't know about them. ये एक अच्छा knowledge होगा आपके लिए. It belongs to us, they are part of self, but somehow we never interacted, our immune system never interacted with them. For example, there is a protein present in the lens which is confined in a capsule. हमारे लेंस में एक प्रोटीन है which is confined in a capsule. When it was first created, it was like created in parallel to our immune cells, something like that. And since the very first time, that protein is enclosed in a chamber and never exposed to the circulatory system and never exposed to any of the immune cells. तो हमारा immune cell कभी उससे मिला ही नहीं है. Now that protein which is part of the human lens, it is a self because it is part of us, but our immune system never able to see that thing. So it can be considered as antigen. अगर हल्का सा भी expose हो गया, it can be considered and recognized as antigens. Those are called as self-antigens or autospecific antigens. There are many proteins which are produced during the embryonic development. And at that time, our immune system is not able to recognize them. They are part of us, or they develop later outside the boundary of the immune system. They can also be considered in the same way. But that's quite interesting कि कुछ चीज हमारे पास है लेकिन हमारा immune system कभी उससे मिला नहीं है. They're called as self-antigens. Next is I need to explain about the cells, the antigen presenting cells and their structures. Any questions? यहाँ तक कोई सवाल हो? I want to explain you about the concept which I talked about. जी शाबाश.
सर मुझे बताएं कि अगर एंटीजेनिसिटी जो है वो पैथोजेनिसिटी और वेरिलेंस से रिलेटेड है तो इसका किस किस्म का रिलेशनशिप है? डायरेक्ट रिलेशन है कि अगर पैथोजेनिसिटी बढ़ती है दैट मींस कि हमारे पास एंटीजेनिसिटी भी बढ़ती है, या फिर उनका इनवर्स रिलेशन है?
ओके, बीइंग ए पैथोजन, एक पैथोजन होना इज अ डिफरेंट थिंग, एक एंटीजन होना इज अ डिफरेंट थिंग. एंटीजन होने से मुराद क्या है? समथिंग which can be detected by our immune system, and our immune system can detect it as a non-self. This is an antigen. Antigen का final level क्या है? If something is detected as an antigen and our immune system can trigger a specific response and can create specific antibodies, then that thing is called as a complete antigen. अभी तक पैथोजन कहीं नहीं आया. Okay. Now, if that antigen is creating trouble in the host system, if that antigen is interfering with the metabolic chemical reactions going on in the system, if that antigen is causing trouble to the cellular functions, that antigen is considered as a disease or a pathogen. But it is equally possible that something which is non-self is not interfering with our cellular system. Something which is non-self, which is an antigen, but not causing a disease, that is not a pathogen. So if an antigen is able to interfere with our cellular functions, that antigen can be considered as bad or the pathogen.